Good morning, students. I hope you are doing well. And the previous classes and all what we have learnt in molecular biology, that is the base for further studies. So to understand the next units, you need to understand thoroughly the DNA, structure of DNA, and the genetic material, why it is called a genetic material or the hereditary material, the experiments on it. Then we spoke about the replication. So these are like base for even biotechnology, even for any further research. So your major work is to read and understand more and more about DNA, why it is called hereditary, how exactly the scientist did the experiment on DNA. All these things we are supposed to know thoroughly. Yesterday I told you some of the other books also you refer to understand the DNA structure and replication. Now let's start with the one new unit. It is actually related to it. It is protein synthesis. So what is protein synthesis then? Proteins, I hope you know it, chemically they are the long chains of amino acid. They are called the polypeptide chains. The unit is called amino acid. So number of amino acids, they are bound to each other with the help of peptide bond. Then they form a long chain. It is called as a protein. So now the question is, proteins are to be synthesized in the cell, right? Why? Because you are having anyway proteins, right? In your diet you will have a lot of different types of proteins. Why you cannot directly use them? Because also when we intake through the oral digestive tract, it will get digested into amino acids. Therefore, once they become amino acids, every cell there will be some amount of amino acids entering. Now, our whole protein synthesis depends upon what kind of protein has to be synthesized by that cell. Means every cell has one particular function to do, right? Suppose you are exposed to sunlight, then your body skin will turn to black color. Why? There is a synthesis of melanin pigment. What is melanin? It is a type of chromoprotein, right? So at that moment of exposure, our cells of the skin have to produce this protein called melanin, right? It is not the same in all the cases, right? If I am talking about the skin, it is keratin. If you talk about the bone, it is ostin protein. If you talk about the cartilage, it is chondrin protein. So types of proteins to be produced will change according to the type of cell, right? Then, as you already know that all the cells are produced from single cell, that was zygote when the fertilization has happened. Now from that zygote, how the cells get differentiated, and after differentiation, how every cell has so many genes, but all the genes are not doing every activity. In some cells, some genes are not working. They are non-functional. In some other genes, some other cells they are active. Therefore, I said, if at all our skin cells produce melanin, that is not the same case with muscles, right? They produce the actin and myosin protein. So, proteins are must for our cell, and moreover, you know that proteins cannot be stored, right? Like carbohydrates, whenever you have carbohydrates, large amount of carbohydrates in liver, this carbohydrate which converts to glucose during digestion, in liver it converts to glycogen with the help of insulin, right? You know this is called glycogenesis. So afterwards, some amount, extra amount of glucose may be converted into fat also if it is in excess. So whenever you are starving, you don't have any carbohydrates, then this glycogen converts to glucose and it's providing you energy, right? This acts as an energy source for your body. But the same thing is not with proteins. Proteins cannot be stored. Once you have proteins, every now and then these proteins will convert to amino acids. Amino acids will undergo deamination through ornithine cycle in liver and form urine, and it is excreted. So whatever protein you are having, it is not stored in any forms. It must be utilized now and then, right? Therefore, your consumption of protein has to be continuous. But that is okay. But what happens in the cell? Every cell, I told you, they will produce protein. How it will do that? That is done by our DNA. DNA contains all the informations required by the cell. Therefore, we call DNA as a master molecule of the cell. It has all the information for proteins and enzymes and hormones, everything it will have. Now, how it will direct the ribosomes? Because the ribosomes are found in the cytoplasm. They are the factories for protein synthesis. Ribosomes are found in the cytoplasm. DNA is found in the nucleus. Now the work of DNA is to communicate with the ribosome and give order that this particular protein has to be synthesized. This is the total story of protein synthesis, right? How it exactly communicates with the ribosomes, how it will produce the protein is the one complete story of protein synthesis. Now, let me give you some information about where and all protein is required by the body, right? You must know the basic thing. Every cell membrane consists of the proteins and lipid bilayers. I hope you know. It's called a bilipid and bilipid layer with the proteins in between. That's called the bilipid protein, lipoproteinous layer. So in every plasma membrane there are a lot of proteins present. So to build anything, to repair system or to build the cells or the cell division, everywhere proteins are required because cell membrane itself consists of the proteins. Apart from that, proteins significance let us study. Why? Because most of the time whenever we write the heading protein synthesis, first you should get the question why it has to be studied. The first thing is proteins are forming the building blocks of our human body, right? Then if you by heart building blocks, it's of no meaning, it's no use. You should have to learn why are they called the building blocks of the body. Because there are certain proteins that give the structural part of our body, like bones, the major skeleton, bones and cartilage. They form the skeletal system, right? They are building our body. But these are produced in turn by certain proteins. So they are called structural proteins. We have various types of proteins. So what are the proteins we have? Right? Proteins, some are called as structural proteins. So these are the proteins that are producing the skeletal system like bones and cartilage. So they are mainly acin that form the bone, chondrin that form the cartilage. Right? Then apart from that, you have keratin that is found on the nails and hair and all. That also is a structural protein, keratin. And some proteins are said to be chromoproteins. What are chromoproteins? They are the colored proteins. They will give certain colors. Like one of the example is hemoglobin. Hemoglobin has the red pigment and hemoglobin gives red color to the RBC, red blood corpuscles. Therefore, hemoglobin is a colored protein. It's called the chromoprotein. Second one is melanin. Melanin gives the black coloration to the skin and hair. Then, apart from this, we have plasma proteins or the protoplast, or we call it as the liquid plasma of the blood, also contain proteins. They are called plasma proteins. These are albumins and globulins. Globulins, right? Then, what are other proteins? All our immune system building components are nothing but proteins. They are called antigens and antibodies. So, immunity, whatever we have, immunity which is built by our antigen-antibody reactions. Antigens and antibodies, they are also proteins, right? These will provide immunity because antigens are the surface proteins of RBC and WBC cells, and they recognize the non-self antigens. Whenever the bacteria or a virus enters our cell, its surface antigen or the protein is identified by our RBC or WBC cells. Then against that particular protein they produce another group of proteins called antibodies. These antibodies go engulf the foreign antigens and they are destroyed by producing lytic enzymes. So this again protein. Then apart from this, I told you a lot of proteins like muscular protein, it is actin and myosin. Then osin, I already told you. And some of the other proteins, what we have is saliva. In saliva also we have some albumin, salivary amylase, all the enzymes. So they are also proteins. So all the enzymes are proteins. That doesn't mean that all proteins are enzymes. Most of the time students will write the characteristic of proteins. You will write all proteins are enzymes. Not that way. All the enzymes are proteins, right? Because proteins are so many types, right? Enzymes also are produced, or they are chemically they are proteins. Then some of the hormones are proteins, right? Like insulin and the glucagon hormones, they are proteinaceous hormones. Apart from that, whatever you have, prostaglandins, testosterone, estrogen, progesterone, they are all the steroidal hormones. But there are some who are all proteinaceous hormones. Okay, so this is why we require proteins for our body. Continuous supply should be there, right? So therefore, if you don't consume proteins at the constant rate, then you may get protein deficiency. Okay, now this is the outside thing. Now let us go to the chemical structure of proteins, right? The chemistry of proteins, you know. As I told you, proteins are made up of units called amino acids, right? Then how are the amino acids combining to form the proteins? Okay. Coming to the chemical structure of proteins, they are consisting of number of amino acids actually. Each amino acid will be like this: CHR, NH2, COOH. Okay, this is one structure of one amino acid. Now, this combines with another amino acid CHR, NH2, COOH. Right? When they are combining, they lose water molecule here, and the resultant structure is CHR, COOH. Here it is NH2. This is NH2, and here it is CO. Okay, this is NH. CONH. This is binding to amino acids CHR, COOH. So this communicating link between the two amino acids is called the peptide bond. It is called the peptide bond. Okay, this is peptide bond. What is peptide bond? It is the link between two amino acids containing CONH group. We call it as peptide linkage. So like that, another amino acid will combine with this another CONH group. Like this, continuously amino acids are combined to form number of one protein will have lot of amino acids, hundreds of amino acids. Now the formation of peptide bond, you have to remember, this may be asked for the linked part. Okay. Now let's move on to next part. How is protein synthesized in the cell? Okay. So what is that actually present is, in every cell I told you, the master molecule is DNA, that is controlling the production of the various proteins. So where are this actually? So if you observe, this is the cell. I just draw one cell, a huge cell I am drawing. This is the cell. Here is the nucleus. Nucleus has nuclear pores, right? These are the nuclear pores, this is the nucleus. This is a nuclear membrane. Okay. This is nuclear membrane. Within this you have number of chromosomes, chromatin and number of chromosomes. Then imagine this is the DNA, double stranded DNA. One DNA possesses number of nucleotides, and its discrete units are called as genes. Maybe from here to here, maybe one gene. One DNA may have large number of genes. Genes are the units of DNA that code for one character or one protein, right? So this DNA part will give the message to the ribosomes to produce required protein. How will it do? It will undergo unwinding first, and once it unwinds like this, its messages, nitrogen based messages that are present over here, that are copied by the messenger RNA one by one nitrogen bases, right? They will be copied according to the messages of the DNA one by one. This is by complementary base pairing they are copied. Okay. Here the unwinding happens and copying happens by RNA polymerase enzyme. This is nothing but the RNA 5' to 3' it elongates, and we call this as the messenger RNA or mRNA. This mRNA carry the message from one of the DNA template to the ribosome through the nuclear pore, right? In ribosomes, this is the ribosome present in the cytoplasm. The mRNA settles and it is converted into proteins, or its messages are translated into proteins. It will have so many messages which are copied from the DNA. 5' to 3' it is read, and here the messages present on mRNA are read 3 at a time. We call that as a codon message, which is triplet codon message. It is having 3 nitrogen bases, so we call it as a triplet coding language. Here this message may be ACG. It is read by another structure, we call it as a tRNA or transfer RNA. It will have opposite nitrogen bases, that is called anticodons, that is UGC, right? Then it will bring particular amino acid which are present in the amino acid pool. There will be a pool of amino acids. From there, this tRNA will pick up the right amino acid and binds to this and translate the message from the nitrogen based message to amino acid message, right? So this process is called translation. The process of transferring of the message of nitrogen base pairs into nitrogen bases into amino acid is called translation. And the first process where DNA transfers its message by complementary base pair copying on the mRNA is called as transcription. So we have two things. One is transcription, another one is translation. So define transcription. How to define it? It is a process of transfer of or copying of nitrogen based messages from one of the DNA template to messenger RNA through complementary base pairing is called transcription. Then what about the next one? That is translation. That is transfer of nitrogen based messages from mRNA to amino acid language through tRNA on ribosome is called translation. Now imagine how the message has transferred. First it was there on DNA, then it transferred to mRNA, messenger RNA, then it transferred to proteins. This complete structure we call it as the central dogma. That is how the message transfers. Central dogma. So DNA on transcription transfer the message to mRNA, and mRNA during translation transfer the message to proteins. Actually DNA to mRNA is not unilateral thing. Sometimes it may be happening reverse also. We call it the reverse transcription. In some of the virus, reverse transcription happens. Once they enter the host, they convert their RNA into DNA. That is called reverse transcription. Transferring of RNA to DNA. It's a rare case, but it happens. Therefore, I am writing forward and backward arrow. DNA converting or transferring the message to mRNA during transcription, and the transcribed message of mRNA transfers to proteins through translation. This is the central dogma of the cell. This is the central controlling unit of the cell, right? Whatever protein is required, this will happen. That is continuous process. So this is what summary of the protein synthesis. This is not the whole story I am giving you. Summary. What is that I am showing? Here is the double-stranded DNA. This is double-stranded DNA. DNA possesses number of units called genes. Genes are the parts of DNA that produce one particular character, which is nothing but protein. Every character is nothing but protein only. So genes are expressed in the form of proteins, right? That's why called the gene expression. Genes are expressed in particular protein. Protein will express in the form of a character. Now this is the DNA having number of discrete units called genes. Every gene has an information which is required for the synthesis of protein. This information must be transferred to the protein synthesizing factories of the cell, that is ribosome. Ribosomes are found in the cytoplasm. They have smaller subunit and larger subunit. This is the ribosome. So these ribosomes get the information when mRNA comes out of the nucleus and settles on ribosome. On ribosome, the messages of mRNA are read in the form of triplet codons. They are called codon messages. 3 nitrogen bases code for 1 amino acid. We call this as a triplet coding language. So this again in detail I will explain you later. Triplet codons we call it. The codons that are read 3 at a time is called triplet codons or triplet coding language. So these 3 nitrogen bases of the mRNA will be read by the transfer RNA. The clover shaped structure is tRNA. Transfer RNA. This tRNA will contain, it will have at its tip the anticodons. Anticodons are the complementary 3 nitrogen bases to the codon message of mRNA. They will read, the anticodons will read the coding messages. If they match, then particular amino acid get translated. Next, another one will be appearing here. Remaining 3 are read and they are bound to form proteins, right? So suppose another one will bind here. Another second message is read by another tRNA, okay, like this. Then its amino acid, this is amino acid number 2, let's say this is amino acid number 5. These 2 are bound by peptide bonds. This we call it as translation. So how do you define translation? It is a process of transfer of nitrogen based language or nitrogen based messages that are brought through mRNA in ribosomes and converting them into amino acid language with the help of transfer RNA or tRNA. That is the definition of translation. This drawing will help you to understand what is transcription, what is translation in short, right? Separately I will be discussing about transcription, which is expected for 5 marks separately, translation for 5 marks. So both are separately asked. Right now you draw this diagram and remember the central dogma. Write down and if possible make a short note of the summary what I explained to you now. Then we will talk separately the separate transcription and translation. Okay. Now let's move on to transcription. Okay students, let us begin with the first process in protein synthesis, that is transcription. So how do you explain or how do you define transcription? It is a process of transfer of nitrogen based messages of DNA, one of the DNA strand, to mRNA through complementary base pairing or copying of the message from one of the DNA template to mRNA. Now how exactly transcription takes place, right? I told you this is the double-stranded DNA. First let me draw the double-stranded DNA. This is a double-stranded DNA and I told you DNA consists of large number of discrete units. These are called genes. This is a double-stranded DNA. You all know this already, you have drawn the diagram and I told you to practice, and next homework will be continuously drawing the diagram of DNA and the replication fork. That will be your homework, because with all the informations you have to draw beautifully and then you have to show me. Okay, so this is the double-stranded DNA. Okay, now it has information from here till here, maybe one gene, from here till here, maybe another gene. So gene is the unit of DNA that will code for particular protein. In turn, protein will give you a character, right? So now imagine information from here. This is a double-stranded DNA. It should have the nitrogen-based messages like this called. Okay, from here till here, inform has to be transferred. Now, how I told you, it has to first unwind. It's not like a replication. The whole DNA will unwind. No, only that part of the DNA that contain the information for particular protein will only unwind. The DNA is so many meter length, the big one. The whole DNA will not unwind. Only that portion that has information for that particular protein will unwind. So that unwinding and copying in the form of RNA will be taking place with the help of the enzyme that's nothing but RNA polymerase enzyme. This is called RNA polymerase enzyme. This RNA polymerase will bind to the initiation site for transcription and go on opening up the DNA like a zipper fashion, right? But this is first I am explaining you initiation of transcription. Okay, now once RNA polymerase has to bind, I told you DNA has multiple informations, millions of information, lot of genes are there. Then how exactly it will bind to the particular gene which is to be synthesized? This particularly that complete enzyme has to bind to the initiation site of transcription. So what is the initiation site for transcription? Initiation site for transcription. The initiation site for transcription will be identified by something called as an initiation factor. Initiation factor for transcription or IF. Its name is actually IF we call it. Its name for any transcription that helps the RNA polymerase to bind the initiation factor. Its name is called sigma factor. Okay. So what happens is the initiation site means from where to start transcribing, unwinding and copying the messages. That is identified by the initiation factor for transcription, which is called sigma factor. Now, sigma factor will identify the starting point. What will that identify? Means what is the speciality of initiation factor for transcription? So, initiation factor will identify the initiation site. Initiation site is normally characterized by presence of multiple Ta base pairs. Right? This is called TAA, AAT, something like this. TTAA, something like this. So, at initial stage of any DNA, there are multiple TATAT or AATTA, like that nitrogen bases are present. And therefore, the initiation site for transcription is popularly called as Tata box. We call this as Tata box because it has TATA base pairs, nothing else connected to the Tata and all. Right? TATA base pairs are the initial nitrogen bases for transcription. Right, where RNA polymerase will bind, and that initiation site is recognized by the initiation factor called sigma factor. Right, this Tata box was first identified by scientist, the Pribnow. Pribnow is the name of scientist. We also call this as a Pribnow box. In eukaryotes, in prokaryotes, in eukaryotes we call it as a Hogness box. Right. So whatever I am writing at the side, you will have to make short 1 or 2 line sentences on that. Okay. So keep that in mind. That will be your homework. What is Tata box? What is Pribnow box? What is Hogness box? Right? And I told you this information. I am going to the deeper explanation. I am not just going to any just NCERT book. Along with that, some extra information I am giving you. Therefore, you need to learn, understand thoroughly, then you write. Listen to me when I am explaining. If you have some other way, you get the information from other way also. Through Google also you can do it. But I want you to write. Means something you work on it. Then only you will understand it. So that's called the Tata box or Ribno box or Hogness box. So now, what is the whole story here I told you? That explain, the whole story explained that RNA polymerase enzyme starts a transcription and binds to the transcription initiation site, and this transcription initiation site is recognized by the initiation factor IF named as sigma factor. It is called sigma factor. We represent like this also, sigma factor. Okay. So it will bind the RNA polymerase to the initiation site for transcription. Now what next? Next, wherever it has bound, it starts moving in this way and start uncoiling the DNA. So let me write how it is elongating. Imagine this is elongating like this. Okay? This is how it is elongating. Okay? This is the DNA, unwound DNA. Now I told you, here will be our RNA polymerase enzyme. Now the polymerase enzyme is here. Now there is no point having initiation factor is done. It has done its work. So initiation factor has left this place. Right? This will not be attached to this anymore. Right? This is the unwound DNA having two strands separated. Now, one by one, these nitrogen bases are copied to mRNA. So, for this one, okay, let me write how exactly it happens later. First, whatever it is copying one here, second one is copied, third one is copied, fourth one is copied, fifth and sixth and so on. This is how mRNA is elongated in 5' to 3' direction. Right? So, how it is copying, that will be asked. So, this I will enlarge and show you. Imagine this is the unwound DNA strand. Okay? Here imagine there is C, there is A, there is T, there is G, T, A. Imagine this way they are there. Here it is G, here it is T, here is A, here is C, here is A, here is T. This is how it has opened up. Now while copying, how it will copy? mRNA will copy in what way? Wherever there is T, then A will come and bind here. So RNA polymerase is here. It is guiding all this. So A will be binding here. For A, U will bind because it's RNA. For C, G will bind. For A, U will bind. Right? They are binding like this. Right? For T, A. For G, C. And so on. This way it is elongating. Okay, it may be A, C, T, U, A, something like that, 5'. So, while transcribing, two things are changed. One, instead of deoxyribose sugar is formed, and instead of thymine, uracil is formed. That's a difference. Otherwise, this question will be asked many a times. It's a very simple thing. They say that if the DNA contain information, so A, C, D, G, something like that, what are the informations present on mRNA? You are supposed to write its complementary nitrogen bases. And it is copying from 5' to 3' direction. Then the copying strand, this should be 3' strand on the DNA. This is a 3' strand from which it is copying. This is a 5' end. One of the strand that is providing information for the mRNA, this is having opposite information to the mRNA. It is called as antisense strand. And the opposite strand, which is here now, this strand of DNA, it has information exactly similar to the mRNA. You can see G is here, G is here. Instead of T it is U, A. Okay, A. That means the information present on mRNA will be exactly similar to the another strand which is not acting as a template. We call this as the sense strand. Sense strand's information is similar to the mRNA information. So this is how the transcription happens. So this way mRNA goes on elongating as the RNA polymerase move further on the DNA. This is double-stranded DNA. It move on the DNA. It keeps on unwinding, copying continues, and it starts rolling again, coiling back. Whichever it has opened, as this copying continues, it will close. Right? This way transcription will continue, and it will end at the end of the gene. Normally, their end of the gene, normally there will be poly A will be there, that is poly T will be there, and mRNA will code for with that. It will be coding with the poly A. Right? So this is the end. Suppose this is the end. There will be termination factor binding to this called as the rho factor. The termination factor is called the rho factor. This will bind and it will recognize poly T sequences on the DNA, and that is why the copying of the mRNA will be at the end of the mRNA there will be poly A. Poly means many. A means adenine. Many adenosine nitrogen bases will bind at the end of mRNA. So that is how the transcription is ended. Now let me summarize. First diagram I gave you, initiation of transcription. Second one is elongation of mRNA. Is an elongation of mRNA. And the third one is termination. This is also elongation A diagram, it is B. Okay. Then termination happens with the termination factor called rho factor, which recognizes poly T sites. So there are 3 steps you are going to write. The last one should be termination. So termination should have terminating factor. Okay. Now, what is the summary of this transcription? Transcription, as you know that, it is the copying of nitrogen-based messages from the one of the DNA template to the messenger RNA with the help of RNA polymerase enzyme. Right? Now, DNA, as it contains large number of informations as genes, one particular gene, if it has to get transcribed, then it has to be bound by RNA polymerase enzyme, which will unwind and copy the messages. But for this unwinding and copying, first it has to bind to the first site, starting part of the gene. The starting part of the gene will bind, it is called as the initiation site for transcription. Then if it binds to the initiation site of transcription, somebody is identifying the initiation site to make the RNA polymerase bind. Right? What is that initiation site? It is characterized by presence of multiple Ta factors. Ta nitrogen bases. We call that is why this is the Tata box. Ta box is nothing but Tata box. This is recognized by initiation factor called sigma factor, and this was first given by Pribnow in prokaryotes and Hognes in eukaryotes. Therefore, we call the initiation site for transcription as Pribnow box in prokaryotes and Hognes box in eukaryotes. Okay. Now, once the initiation factor, sigma factor, recognizes the initiation site, it binds the RNA polymerase enzyme to that initiation site. That's done. Now, next is work of RNA polymerase enzyme. It starts moving on the DNA in this direction. While moving, this sigma factor comes out. During that process, it will unwind the DNA. As the nitrogen bases are opened up, one of the strand acts as a template, which is normally 3' end strand, act as a template and provide the information for the formation of messenger RNA. What happens is, this copying will happen in complementary base pair fashion. That is, as the nitrogen bases are opened in the template is T, the copied message on the mRNA is A. ATGC pairing, you already know, base pair pattern only it will happen. If it is again A, then instead of thymine, AT I told you, but it is forming RNA. Therefore, there is no thymine in RNA. Therefore, instead of thymine, uracil will bind. And if the template has C, then the complement strand of the mRNA will have G. Then, if the template has adenine, then it will have uracil. Again thymine, adenine, guanine, cytosine, and so on. That's how mRNA get elongated. It becomes a long strand of mRNA till the end of the DNA, the gene, it will be copying the messages. Now, the strand which is providing information for the synthesis of mRNA will have opposite nitrogen bases or complementary bases, therefore it is called antisense strand. Whereas opposite strand that is not forming the template, it is not providing the information for mRNA, but its nitrogen base messages are similar to the mRNA messages, we call it as a sense strand, right? That's how we have to identify, study that. Then after the formation of mRNA, at the end of that particular sequence of nitrogen bases, at the end of the gene, it must stop transcribing. That time there will be a termination factor that will be called as, that is called as the rho factor, will bind to the RNA polymerase and it will recognize poly T sequences on DNA. Therefore, the end sequences on the mRNA are poly A. That's how it will recognize and the mRNA is released. mRNA is, means it will get unwound and it will come out, right? So, rho factor recognizes the termination factor. That's about the formation of mRNA, that is transcription, right? So, remember properly in this whole thing I explained you. First, protein synthesis, why it will happen, although we are taking proteins, why the protein has to be synthesized. Second one, the protein requirement, significance of proteins by the cell. Protein is required because it forms our cell membrane, it forms our mucin, it forms our cartilage, it forms our bone, it forms our immune system, it forms our hemoglobin, everything, enzymes and hormones, right? Therefore, proteins are required. Then, how these proteins are synthesized? They have to be synthesized in the cell. Before that, what proteins contain chemically? Proteins chemically made up of number of amino acids. These amino acids are bound to each other by peptide bond. So, amino acid, how they bind together by peptide linkage is by forming COOH group. Already I told you, COOH, from that OH is lost, from NH to H is lost, they form water and CONH forms a peptide linkage that forms a long chain of polypeptides. That is the summary of the protein chemistry. Next, after protein, how is protein? We will have to learn about where it is produced. Within the cell, we have nucleus. In the nucleus, we have chromosome. Chromosome contains long strands of DNA. Then DNA normally is consisting of huge number of genes. Genes are the units of DNA that are going to code for particular protein. Protein in turn giving rise to a character. Therefore, gene is expressed in the form of proteins. This is called gene expression. Gene is expressed in the form of proteins. Now, how exactly these proteins are produced in the cell? The information for any protein will be present on the DNA in genes. Then, this gene present on the nucleus has to take the information to the protein synthesizing factories, that is in ribosomes. Ribosomes are in the cytoplasm. Then, who will take the information? The information from DNA's one strand will be copied to the messenger RNA, which is called messenger because the message is transferring the message from the DNA. One of the strand it copies by complementary base pairing and transports to the ribosomes. While copying, it will do complementary base pairing. This process we call it as transcription. Once the transcribed mRNA enters the ribosomes, it will settle on the ribosome and the nitrogen based messages present on the mRNA are called code languages. They cannot be recognized because they are in the form of nitrogen bases. This coding language of mRNA will be translated with the help of transfer RNA or the tRNA. tRNA is nothing but transfer RNA. Transfer RNA is a clover-leaf-like structure. Again, in the future, we will be explaining you the structure of tRNA. But right now, you keep it in your mind that tRNA is like a clover leaf having 3 loop-like structures. This tRNA will recognize the coded message of mRNA with the help of its anticodons. They are complementary to codons, and codons always are read 3 at a time. That is why we call it the triplet coding language. Now, 3 nitrogen bases of mRNA are read by anticodons of the tRNA. If they bind, that tRNA will bring particular amino acid according to the codon present on the mRNA, and messages translated from codon language to the nitrogen based language to the amino acid language. It is called translation. Right? Now, transcription details are given you. You need to draw this diagram and write some points on this. Okay? This is your work. Again, I told you, your homework is DNA, structure of DNA you draw clearly, then write some points on that, on minimum 6 to 10 points. Then replication of DNA, draw the replication code and write points, and draw this transcription, 1 or 2 lines explanation for each of the steps. Okay? Write this. Okay, did you understand now? So, as I told you, I have given you homework. You better write down the homework, what all I told you. The 1 mark questions, you list them off. I am listing it. Just remember, first one in this, what is the peptide bond? Okay, how peptide bond is formed. Third is define transcription. Fourth is define translation. 2 mark question. What is central dogma, right? And how exactly information from DNA flows to proteins is called central dogma. DNA on transcription transfer the information to mRNA, mRNA on translation transfer the message to protein. That is called the central dogma. And I told you, even DNA transferring to mRNA is not unidirectional. It is sometimes bidirectional. In some of the virus, this information of mRNA will be, they will have only RNA as a genetic material. That RNA will be converted to double-stranded DNA by reverse transcription. Right? That's a reverse transcription where it converts to the DNA. Therefore, I told you DNA with 2 forward and backwards arrow marks I showed you. DNA to mRNA or mRNA to DNA. DNA is called reverse transcription. It's a rare case, but their normal ways DNA to mRNA transcription only happens. Right? Then RNA, mRNA form will be transferred to the proteins through translation. That is the central dogma. Right? So, writing this is now your work. Right? Finish this. Then write those points, whatever I asked you, and then I expect you to show me the answers by tomorrow. Okay? Whatever I explain you translation, that we can keep it for the next test or next homework. Okay? Just write them down. Come to the next process, that is translation. So once you have finished with transcription, now you have to think that the mRNA has come out of the nucleus. So before it translates, there are a lot of changes happening in this. So what is that actually? So mRNA structure. So I am not explaining translation now. Let me give you something about mRNA. So mRNA has all the informations like this. Okay. In this information, in eukaryotes, there are some units that are not coding. Some of them are coding, some are non-coding sequences. So these coding languages are called as exons. And suppose in between there are something like this, they are non-coding ones, they are called as introns. So there are a lot of introns that are found between the exons. There is another intron here between so many exons. What are the exons and introns? That is the 2 mark question, distinguish between exons and introns. So exons are the coding sequences of mRNA, introns are the non-coding sequences of mRNA. So before translation, there is exonuclease activity in which all these introns are cut and removed and joining the exons. So exons are only joined, these are the coding sequences. Coding sequences join, and this is called purification of mRNA. Purification of mRNA. mRNA gets purified and the long mRNA cuts into short one. And will be having one kind methylated end one side you have poly A end. With that, this is ready for getting translated, because unnecessary informations which are not required for or which are non-coding sequences need not be translated at all. Therefore, in eukaryotes, these non-coding sequences or the introns are removed and cut and removed and joining the exons. Okay. Now, what happens exactly in next thing? How exactly this complete information of mRNA is translated to proteins? Okay. This you understand thoroughly, then almost the molecular biology ends here. Because once molecular biology, if I say the basics of molecular biology, all these things are basic of molecular biology. One is DNA, one is the experiment on DNA, next is replication, next comes your protein synthesis. So you need to understand protein synthesis thoroughly. That's end of it. Later, almost the things are depending on this. Basically, you have to have this. Based on this, only other things are built. Okay. Now come to the translation. The mRNA. Imagine this is mRNA having all the information on it. It has entered the cytoplasm. Okay. From 5' to 3' I am writing here. It has coded informations on this. Right. These are the coded informations. These are called as triplet coding languages. These are the nitrogen based messages brought by the mRNA, which were copied by DNA during transcription. So what I am writing is, these are all the coded informations of mRNA. Okay. These informations are in the form of nitrogen bases. Right. Starting few you are leaving, and here the coding languages start. Here 3 of these codons together they form a triplet codon language. This is a triplet codon. So they will have anything like A, U, G, C, C, A, anything it will be, C, A, G, A, anything like this, G, A, C or something like that, right? C, A, C or something like this, okay? This will continue. So 3 nitrogen bases will be coding for one amino acid. This is called as the triplet coding language, right? Now, this information carrying mRNA has entered into the cytoplasm. This is mRNA. Once mRNA enters into the cytoplasm, some activities happen in the cytoplasm. First activity is activation of amino acids. Amino acids. So amino acids are there. This is in the cytoplasm. I told you, this cell. This is in cytoplasm. All the cytoplasm, they will be having number of amino acids in the amino acid pool. These are all amino acids. These amino acids take up an ATP molecule, and in presence of enzyme, that is amino acyl synthetase, amino acyl synthetase enzyme, they will get activated amino acid. Activated amino acids. Okay, this become activated amino acids. So once they get energy, they get activated. So first step is activation of amino acids in presence of enzyme amino acyl synthetase. Now this is mRNA. These are the coded messages. Any message will be there, A, C, A, something like that. So starting normally you will have AU, right? AU, because it is copied from Tata box. So it should be TA means UA will be copied, right? Therefore, the starting ones are like this. Now, how exactly translation begins? Once mRNA enters into the cytoplasm, it will settle on the ribosome. So ribosomes, before protein synthesis, they will be separated from each other. Normally, a ribosome, if you observe, the structure is like this. One smaller unit will be there, one larger subunit will be there. You must be knowing. In prokaryotes, ribosomes are, so prokaryotic ribosomes are, how are they? Prokaryotic ribosomes are of 70S type, and their subunits are 50S and 30S. In eukaryotes, how is the ribosome? In eukaryotes, ribosomes are 80S type with the subunits 60S and 40S. Okay, so that is the ribosome. So ribosome, complete ribosome has 2 subunits. One is smaller subunit, one is the larger subunit. Now coming to the ribosomal subunits, the smaller subunit, normally whenever there is no protein synthesis, they will not be together. They are separated from each other. So First mRNA settles on the smaller subunit of ribosome. Okay, this is the ribosomal smaller subunit, right? Like this is the ribosomal smaller subunit. I have drawn it. Yeah, larger subunit is not there here. Okay. Now, next step after the ribosome, the binding of mRNA with the ribosome happens. Not the whole ribosome, only smaller subunit of ribosome. Another action happens, that is, there are a lot of rRNA, the tRNAs are present in the cytoplasm, right? Here you find number of tRNAs. This is tRNA, this is tRNA. So here I am drawing them clover leaf-like structure. They are called as the tRNAs. They are having some anticodons at the tip, CAG, UAC, something like this. They are the tRNAs found in the cytoplasm. These tRNAs will go and bind or they will catch the amino acids. So that is the second step of protein synthesis. What is the second step I said? That tRNA get bound with the activated amino acids. That process is called as the formation of tRNA amino acid complex, right? Or we call it as tRNA, amino acyl tRNA. This is otherwise called as amino acyl tRNA. T always should write smaller one. So what happens is the right amino acid will bind to the type of tRNA based on its anticodon. There are totally 20 types of amino acids in human beings. As I started with the initial explanation, I told you that amino acids are the units of proteins. Amino acids are of two types. One is essential amino acids, some are non-essential amino acids. Essential amino acids needs to be taken through the diet. Means certain amino acids you need to consume them orally. Some of them are synthesized in the body itself. They are called non-essential amino acids. Now, these are totally 20 types of amino acids in every cell. Only 20 types we have amino acids. So, these amino acids, once they get activated, they have the ability to bind to the tRNAs 3' end. This is a 5' end, this is 3'. 5' end, 3' end. These will bind to the tRNA to form amino acyl tRNA or tRNA amino acid complex. Right? Now they are ready to come and bind to this. They are anticodons. These have codon. They have anticodons. But what happens is, to start with, always initiation of translation happens by one of the codon. So the first codon in this is called as the initiator codon. Initiator codon on the mRNA is always AUG. This codes for the amino acid methionine. Right? The first one always should be translated to be AUG. It is called the initiator codon. Right? It will be coded by the amino acid methionine. So this is the first one. So if it is a eukaryote, it is methionine. In prokaryotes, it is called as formal methionine. Methionine, or we call it as a F-methionine. Okay. Now, this I will draw again another diagram of tRNA that carries it. Right. This tRNA, UAC are written. I will just draw it slightly bigger one. Okay. Here is UAC, and here is the binding of the methionine. We call this as met tRNA or methionated tRNA. This is going to come at the beginning. Okay. So remember, this is the first one to enter for beginning the translation. So this has come all the way here and it has translated this message. Okay. This is the translation happening. It is U, it's A, it's C. This is translation beginning, and here at the tip we have methionine. We call it the met tRNA. Draw this diagram first. Okay. I hope you will be drawing it. See the translation diagram. I hope you have copied this. This actually possesses some important things in it. This is mRNA carrying this. I told you the initiation happens with met tRNA. What is met tRNA? Met tRNA refers to methylated tRNA. Here, first thing you have to remember is always the first triplet codon to be translated is the AUG. AUG is coded by the initiator protein, the amino acid, that is methionine. In eukaryotes, it's called methionine. In prokaryotes, we call it as F-methionine or the formyl methionine. Now, as first codon to be translated is only AUG, there will be formation of met tRNA for the first time. This is called as methylated tRNA or met tRNA. This met tRNA comes to the point where it has to translate. It will come this way and it will bind to this. And in prokaryotes we call it F-met tRNA. This will bind, you can see here, it has bound AUG, UAC. They are binding here. Okay. This is met tRNA. So it has initiated the process of translation by binding to the initiation site for translation. That is AUG initiator codon is there. Now, once it binds, there is a time for the larger subunit of ribosome to bind to this smaller subunit, forming a ribosomal complex. This is the ribosomal complex, right? This is the complete ribosomal complex. So I showed you step by step. First one, only smaller subunit will be there. To this our mRNA will bind, and after that the activation of amino acid happens. Then all the tRNA get bound with the amino acids, forming amino acid tRNA or tRNA amino acid complex. But apart from that, the first one, there is a rule in this. That cannot be anything, it should be only AUG. The first nitrogen base codon to be translated is AUG. We call AUG as initiator codon. So to this AUG, UAC should be the anticodon on tRNA. So the anticodon UAC possessing tRNA get bound with the first amino acid, that is methionine. Methionine is the first amino acid binding to initiate the translation. And here methionine is found in eukaryotes. In prokaryotes it is formyl methionine. And such tRNA we call it as the F-met tRNA in prokaryotes, met tRNA in eukaryotes. So such tRNA will enter to the site of ribosome, binds to the AUG with the UAC, then it will form first translating unit. So see, three things should be matching. Here should be the amino acid, here is the tRNA's anticode, here is a codon, right? At the 5' to 3' end, methionine will bind, or amino acids are bound to the tRNA. Now, next codon is CCA. You can see here, CCA is the next codon. So after the first codon, there is no compulsion that this code only should be there. It can be any codon, but starting codon only should be AUG. Therefore, the next codon can be anything, right? So as is written here, CCA. Then what kind of anticodon you are expecting? What kind of tRNA you are expecting? So this is the next tRNA entry. So I will just remove these diagrams, because again it may confuse you, because it's the next story I am giving you. So this one is the ribosome now. Now the next tRNA which brings the amino acid is, this is the next tRNA, its anticodon loop, other loops, this is the amino acid. Amino acid can be anything, 10th, 11th, 12th, anything. It may be serine, it may be glycine, it may be tryptophan, anything. Then here GGU it is bringing, GGU, that is binding, because it is CCA. CCA anticodon should be GGU, right? Now, this is the ribosomal complex. This is a full ribosome. This is the smaller subunit of ribosome. Let me label this side, because I don't know whether you can see it. This is the larger subunit of ribosome. This is the smaller subunit of ribosome. Together it is the ribosomal complex. Right? Now, once the ribosomal complex is there, then the second one will come here. Now, coming to this ribosomal complex, this has the larger subunit possess two places. See, these things are taken down, I will just explain you here. This larger subunit of ribosome possesses two units or two areas for the binding of the tRNA with amino acid. The first area, imagine this is the ribosome. Okay, the first area to which it binds is called as the A site. Second one is called as the P site, and third one is called as the E site. We have three sites, right? So where I have written the second one, this is the A site, this is the P site, and the third one here it will be E site. Three sites are there: A site, P site, and E site. What is the meaning of A site? A site is called acceptor site or amino acyl site or acceptor site. Then P site is called polypeptide site. Polypeptide site. E site is called exit site. So there are three sites on the larger subunit for the binding of the tRNA. First is A site, that is amino acyl site or the acceptor site. P site, it is the polypeptide site. Then last one is the E site or the exit site. Now what happens here? So once it has started, already there was a first amino acid formed. Now both are filled. Here A site will be occupied by the new amino acid tRNA. Then what happens is now this whole ribosome start moving on the mRNA, leaving the first triplet codon, it moved to the second triplet codon and reads the, see, this is the first codon, codon number 1, this is the first codon, okay, this is the first codon, this is the second codon, this is the third codon, this is the fourth codon. So it leaves the first codon and go to the second codon, right? So that diagram I draw separately now. The same diagram will not hold good now, right? Keeping the types of sites in mind, let us continue with the next diagram. This is diagram 1. This diagram explains you initiation of peptide bond. Now let me tell you where is the peptide bond. Okay. So suppose this is the first one which has brought methionine. Second is any amino acid has come. When the two sites are full on the tRNA, these two get bound with the peptide bond. This is the peptide bond. Peptide bond is formed already. Okay. This is initiation of peptide. This diagram gives you how peptide bond is initiated. Now it will elongate. How? The whole of ribosome. Imagine this ribosome is from here to here. This is a ribosome, smaller and larger subunit. This will move to the next one. Okay. Let me draw the second diagram over here. Okay. Leaving the first ones. I am leaving the first ones. Imagine all these things I have recorded. It will move to the third place here. Imagine it is CAG written. Let me write CAG. Its previous one was CCA. Here it is CCA. Before this it was AUG. Right? Now what will happen? The whole ribosome shifts from the first codon, this is the first codon, the second codon, this is the third codon. It will shift, leaving this and go to the next codon. Here is our smaller subunit now. Here is our larger subunit of ribosome. I hope you understood now. Right? This is the first codon is left out. Then where are our tRNAs? So the whole ribosome is moving. Therefore, the tRNA present at A site will shift to P site. The one which is present at P site will be going to the exit site and will come out of this. Right? So now let me write this is the first tRNA. This is the second tRNA. Now first tRNA will go out. The second tRNA come to the P site. So here I told you there are two sites. This is the A site. This is the P site. This is the exit site. Okay. Now the second tRNA has come over here. Okay? The second tRNA having GGU, and now it has amino acid 10, I told you. Then the another one, that first tRNA will go to the exit side. It is here now. After it leave the place, it will be going out. Its tRNA, its amino acid remain attached to the tip of this. That's the methionine. Right? So they have already attached. What happens? It will separate and this will remain on the top of this. Right? Therefore it's come here. Now this is leaving the place, it is exited. One of the tRNA which had come first. So there are three now. First one, second one, third one. When the third one arrives, first one exits. Then the second tRNA will go to the P site. A site become free. Now A site is free because it has entered to, this is the second one. First one has gone. Now the third side, the third triplet codon is read by another new tRNA. New tRNA has the information anticodons for this. It is GUC. Then it has got some other amino acid number 5. Right? Once this binds to the A site, this is P site. A site and P site, the amino acids are bound by peptide bond. This is one peptide bond, the second peptide bond. Okay? Now, another one, next story. This is first one is gone. The first tRNA has left the place because its work is done. It has got the amino acid, it has brought to the tRNA, it has brought through the tRNA and left it in the ribosome by binding it to the another amino acid which is there in the P site. It has left now. Now, next story. The next information is written as CAG. Okay? Imagine CAG is red now. That is red. Next, this is third one. Fourth one will be red. AAA I have written. So there is AAA. This is AAA. Now what will happen? Imagine the whole of the ribosome will shift to the next fourth codon like this, and the one which was second tRNA, this is the third tRNA. Second tRNA will go to exit site and comes out. And now the third tRNA will go to the P site and the new place will be occupied by the, it will go to the A site which is empty, new tRNA enters. Right? I hope you understood. This is like, you should visualize this. Right? Imagine there is a first, this is the P site, here is the A site. Right? Only two people can fit. Exit site is the other side. Exit door it is like. Right? First, this is the ribosome. There is one person in B site, one person in A site. Right? Then what happens once the P site and A site people are full? The person, that's a tRNA present at P site, will hand over. So the one they will bind with the A site and forming a peptide bond. Their ligages or amino acid, they are bound with the peptide bond. Right? Once they are bound, the one at P site will exit, it will go out, and already its amino acid is there on the one we were having who was in A site. Now the tRNA present A site shifts to the P site. Now what is free? A site is free. Now it is occupied by the new amino acyl tRNA complex which comes to the next point, that is A site. Then again, next story continues. The one which comes to the A site, its amino acid binds to the amino acid present on the tRNA at P site. The two get bound by peptide bond. Okay? Now, next thing is, one at P site will exit. Its tRNA will be carrying all the three amino acids. Now, the one at A site comes to the P site. A site is free. New amino acid comes here. Again, the two are bound by peptide bond. Then, the one at P site will exit. Then, A site tRNA comes to the P site and A site becomes free. This is a continuous process. Imagine like that, continuously the amino acids remain bound like this. Okay? These are all amino acids. This is the elongation of polypeptide chain. Therefore, we call this as the P site, means the polypeptide site. It is forming polypeptide bond. This is a polypeptide site. A is the acceptor site. It is accepting. Acceptor or amino acyl site. This is the exit site. That's how this is the elongation. This is elongated. This is called the polypeptide chain. This is a chain. Chain of polypeptide is nothing but the protein. This way it goes on elongating the polypeptide chain, and at the end it will continue till the end of mRNA from 5' end to 3' end. If at the end any codon, namely UAA, UAG or UCA, UAA, UAG, UAC come at the end, these are not coding for any of the amino acids. These are not recognized. If any codon named UAA, imagine UAA comes here, then no tRNA can read this, no amino acid can bind to this. That's an end. These are going to stop the protein synthesis or translation, and these are called as the stop codons, or they are called as terminator codons. They end the process of protein synthesis. Any one at the end, normally they will be present at the end, they stop or they terminate the process of protein synthesis. That's how translation is completed. So we have steps. I told you. What are the steps? Just draw the diagram first. So two diagrams are required. This is the first diagram which is showing initiation of peptide bond. This is the second diagram. This diagram is written as, the heading is elongation of polypeptide chain. Polypeptide chain. So you need to draw two diagrams for the process of translation. This also with another cell. Another cell. This is one cell. That is another cell. Right? Let us finish this diagram. Then we will talk about the summary. Okay. See you. Red one.